Hi, this is Cecilia from the Mango Lab. Our lab belongs in the pharmacy department of USC. And as for myself, I'm a sophomore year student in quantitative biology in the Dornside College of USC. For my interest in bioinformatics, it starts from high school because in middle school, I start learning computer programming. And then in high school, I took more courses in biology. Therefore, I found these two fields are very intriguing to me, and I decided to take the combination between these two as my research directions. Therefore, after entering USC, I take quantitative biology as my major and start doing bioinformatic researches in the Mango Lab. My research topic is the assessment of reproducibility of small uncomputational replicates. In recent years, bioinformatics rapidly evolves, therefore, there are various new approaches and algorithms coming out. And one important way for us to make evaluation among all these tools is to assess their reproducibility. In the past, the common way to make this assessment is by generating the biological replicates, which can be costly and time consuming for most of the time. Therefore, in this project, we use computational replicates instead to make the assessment of reproducibility on SMALT, which is a DNA sequencing alignment tool. As for DNA sequencing alignment, it stands for to find the original location where the DNA reads are originated from on the reference genome. For the reference genome, we choose GRCH38 from NCBI, and we took 10 samples randomly selected from whole genome sequencing dataset. Then we use two computational techniques to modify these 10 samples into four versions. The first is the reverse complement, and the other three are the shuffle sequences version. For the first step, we build index from the reference genome, and then we map these 50 sequencing reads onto the index. For mapping, we use -R to control that the multiple alignment pairs are reported as not mapped because only primary alignment reads are considered in this research, which we referred as the unambiguous reads. Because in our project, since the primary alignment read stands for reads that has been found in only single location on the reference genome, it's easier for us to make the comparison about the consistency in the results. So as for our results, we see that the maps of the reverse complement reads are largely shifted compared with the map generated from the original reads. There are about 1% of reads mapped on different locations on the same chromosomes, and then about 0.5% of reads that has been mapped on one chromosomes in the original reads, but have been mapped on different chromosomes on the reverse complement reads. But this kind of inconsistency did not occur in the shuffle versions. In the graph below, we could also see that the blue portion stands for the portion that is completely identical between the computational replicated version and the original version. So we can see that for reverse complement version, uh, there are a portion that has other color. For instance, this purple portion that is relatively large in the other color area is stands for the inconsistent type, which has the reads mapped only on the original reads, but is not found in the reverse complement reads. In our conclusion, the algorithm of SMALT has a great reproducibility in shuffle reads, but is not very compatible with the reverse complement reads. Therefore, we recommend future researchers to stop using SMALT on research that involves with reverse complement reads because the computational replicates is already an optimized situation compared with the clinical data. So in clinical settings, the inconsistency might be even worse. Um, as for our future direction, we will start to applying the assessment on other kinds of DNA and RNA alignment tools and generate a more wholesome comparison of consistency between different tools.